I'm Caleb Burris. It is February 5th, 2023. For my interaction between a prokaryotic and a eukaryotic cell, I chose the interaction between Shiga toxin-producing E. coli, or STEC, and human intestinal epithelial cells. I did all my drawings through PowerPoint because my handwriting is really bad, and I feel like it would be easier for you to read and for others to read if I was able to produce these through just using shapes and stuff from PowerPoint, but I did all these myself. So on this slide, we see the human epithelial or intestinal epithelial cell on the right side and the E. coli cell on the bottom left. I'm not going to cover everything on the slide. I just want to show you some important things that are going to be important for their interaction. On the top right, we have the cilia cells up here, or part of the cell. And what these do is these increase the surface area for more absorption for these intestinal cells. And then we also see in red and yellow down here, we see the rough ER, and then the red is the ribosomes. And then for the bacteria cell, or E. coli cell, we're going to see one major difference here. We're going to see, of course, is there 3 layers of their shells. So we have capsule, cell wall, and plasma membrane. But what's important for their interaction is going to be these pili and these ribosomes in here. Okay, and so last thing I want to point out here is their size difference. So we see that the epithelial cell is about 8 micrometers, and then the E. coli is about 4 times less, which is about 2 micrometers. So with the interaction, the first thing that's going to happen is these E. coli are going to get rid of some of these cilia, or these microvilli, on the epithelial cells. This gives it a spot to land. And once it lands, it's going to insert a syringe-like apparatus into the epithelial cell so it can start pumping in some important things that start doing work on this cell. We see there's 2 parts to this. There's the blue part and this pink part here. Okay, and so what happens is it starts pumping in these receptors. Specifically, we're going to talk about the translocated intimin receptors. And what these do is it really kind of latches it down onto this epithelial cell, so it's not going to go anywhere, right? That's in pink right here. These little pink arms on step 2. And then once it's latched on there, it's going to start building. And so what I mean by that is it's going to, on step 3 here, it is going to start recruiting some substrates in the cell that will start bringing in actin, because these substrates, what they do is they rebuild the cytoskeleton whenever it needs to happen. And so the bacteria is going to use this in its favor and start recruiting a bunch of actin, more actin than it probably needs. And so what happens is it's going to start bulging the cell. So we get this little uplifted here underneath our bacteria cell. We get this bulge from all this actin that's in there, right? And so that it creates a pedestal is what they call it. It's a little pedestal for this E. coli to sit on. Okay, and then at some point we'll talk about what's going to happen next, but it's going to be bulging a little too much. Okay, and at some point, as it keeps recruiting more and more, and then it starts, it's going to start going to lysis. Okay, and so lysis is just cell death for this bacteria. It's going to explode. The reason this happens, or at some point when this is happening, Shiga toxins start getting produced. Okay, and that's these little yellow clouds around my big red cloud here. Okay, so this big red cloud shows that the bacteria cell has exploded, and then my yellow cells here are my Shiga toxins that were produced before it lysed. Okay, and so once these Shiga toxins get expelled to the extracellular matrix, it's going to try to go back into the cell, and the way it does this is through endocytosis. So the host cell will absorb some of these Shiga toxins, and then the vacuoles or vesicles, will start bringing these towards the ribosomes and the rough ER, and as well as the Golgi apparatus. But I really just focus on the ribosomes for this interaction. Okay, and so once it hits those ribosomes, okay, it's going to inactivate that 60S subunit, okay, of these ribosomes. So that means that we no longer can have a specific tRNA binding to these ribosomes, which means we can no longer have these long polypeptide chains, which is going to lead to the loss of function of the epithelial cell. At some point, it's going to lead to apoptosis or cell death. Okay, so how do we get this? So we get this from cattle. Okay, so usually these E. coli mature in the cattle's guts, and then at some point it's going to poop out this E. coli, and these fecal matter can get into our food, meat, milk, dairy products specifically, and then we eat those and we get E. coli. And so what E. coli does to the human is it's going to, it does many things, but one of the specific ones I'm going to focus on is diarrhea. Okay, so it causes diarrhea because it causes these epithelial cells to lose function, so they no longer are absorbing water, and then they will keep dying by apoptosis and getting rid of all this water as well. So it's not absorbing it, it's going to be letting it out as well. It's going to lead to diarrhea, which can lead to major water loss. Okay, and it also does a bunch of other things all over the body, but like I said, I'm just focused on human epithelial cells in the intestine. And that is the E. coli's interaction with our intestinal cells.